Good evening, ladies and gentlemen, and welcome to this installment of How to Best Study for Dr. Lawson's Lectures. Today we are talking about the DNA Technology lecture, and we will go through all 3 hours in this one video. A key part of what you need to know for this lecture is that you need to know how to visualize DNA. You must always do something to DNA to make it visible to the eye. We either put proteins or dyes on it. We can cut it before we dye it with restriction enzymes or other methods, and we can copy it using dyes attached to it. All of those allow us to see it. But in any case, you always end up with a protein or a dye attached to the DNA so that you can see the DNA. You need to know that gel electrophoresis separates DNA on the basis of size. The top of the gel will always be the region that is the longest strands of DNA, and the smallest will be at the bottom of the gel, or the lower part of the image. We then take the DNA after we've run it on an electrophoresis, and we can immobilize it and then visualize it by things called blotting techniques. You do need to know at least these 2 blotting techniques here, southern blotting and northern blotting. You do not need to know western blotting for this lecture. When we put dyes on DNA, you need to know that what we normally do is we put the dyes on the DNA so that we can see them. For example, in a metaphase squash of DNA, as we see here in this slide, this is a more traditional method of staining the DNA so we can see it. The DNA has been dropped into a single droplet under a microscope slide. They actually come out looking like they do in B. Then we cut each of them out and line them up so that they look like this image that you're more familiar with from your textbook images of the 23 chromosomes. When we line them up, we use the banding pattern that's caused by the dye, typically the dye DAPI or some other kind of staining method that will allow us to see these banding patterns that are very specific for each of the chromosomes, and allows us to know exactly which chromosome is which. We can also now put fluorescent probes. These are little pieces of DNA that have homology, or they bind to, by base pairing, the regions of a certain region of the gene. And so, for example, this fragment here is using the telomeric region of chromosome 8 so that we can actually see that section. We can also do it where instead of doing this black and white type banding pattern, we can just visualize the overall image of the DNA, and then we lay the fluorescent probes over the top of it so that these probes are, of course, base pairing to the DNA itself, but they have different coloring to tell us different things. So the probe here in pink is actually telling us where the centromere is on this chromosome. The part in green is telling us where the subtelomeric region is. We've used very specific probes to see those very specific regions. And you can see in C, D, E, and F that we can do variations of that probing to look at variations in exactly what is next to what, and where, for example, a chromosomal break has occurred. We can also do this last technique that's seen in F. This is called chromosome painting. Chromosome painting, of course, is when we use many probes that cover large regions of the DNA, and we cover it so that each region has a different unique color. This is called DNA painting. Remember that there are 4 kinds of blotting techniques that we need to know about. You need to know how to read a southern blot. You need to know how to read a dot blot. You need to know how to recognize a northern blot. A northern blot is always using patient RNA with a probe that's either synthetic or cloned DNA. The southern blot always uses patient DNA and then probe it with synthetic or probe DNA. Dot blots usually use the DNA. They can use cDNAs or RNAs, but typically it is DNA. So you can use whichever you need to use in that particular case for the patient. Remember that microarrays are just like dot blots, except we can look at a whole bunch of different genes simultaneously. The key that you need to know about Southern blot is that you need to know that the restriction enzyme cutting sites, which are indicated by these down arrows, vary between different alleles in the population. And the probe is a region of DNA that hybridizes to both sides of these two fragments. It only hybridizes to the 6.7 fragment here and the 5.7 fragment. Basically, it's hybridizing to the regions directly under where the probe is drawn. You also need to be able to read a Southern blot. You need to be able to read a Southern blot. I'm going to show you how to do that with this one because we used a different Southern blot when we used in class. So let's look at this one. We have a mother and a father, and we need to figure out where each child got their various alleles, whether they got their alleles from their mother or they got their alleles from their father, and if all of these children belong to both of these parents. So this child does not have the upper band that the father has, so its lower two bands do match the father, so that must be the alleles they got from their father. The mother of this family does not, however, have those lower two bands. That suggests that she is not the mother because she can only give one of these upper two bands, and she doesn't give either one of them to this child. To child 2, you can see that the upper band must come from the father because he cannot give this 6.7 band, but the mother can. So this child could possibly be the child of both this father and this mother. The third child has the upper band that is like both parents. We can figure out that it comes from the mother because the father is the only one who can give the 3.9 and 3.6 bands. So these lower two bands are from this top allele provided by the father, and the upper 7.5 kilobase allele must come from the mother. Likewise with child 4, the lower two bands must come from the father, and the upper band must come from the mother. Make sure that you can read a Southern blot and assign where each person in the pedigree received their DNA bands. Do remember in Southern blots that there are always two alleles. For example, this child is homozygous for this allele configuration. It has 2 copies of the 3.9 band and 2 copies of the 3.6 band. You can't tell the difference because they're the same size. Remember, in Southern blotting, what you are being asked is what size is the DNA and is the DNA gene there. So Southern blot tells us both size and amount of DNA there perhaps, but more importantly, is it there, yes or no? Dot blots, on the other hand, are telling us that the DNA is present or the RNA is present. That's all it's telling us. It gives us no indication about size, but it does tell us yes or no, the gene is there. Microarrays work exactly the same way. They just tell you a yes or no, we have the complementary DNA or RNA present. In Northern blots, of course, it's more like a Southern blot. It's telling you both size and yes or no. In DNA sequencing, remember that in DNA sequencing, we take a primer and we extend it because remember, all DNA polymerases need a 3' hydroxyl. We extend the DNA in the presence of dideoxynucleotides. These dideoxynucleotides then terminate the progression, and we get a collection of all the places where, for example, in the blue test tube, where the dideoxy G is added. And then we can run those out on a gel electrophoresis, and we can actually see the size separation. and so that we can read the sequence. You will never have to read a sequencing gel. This is just so you know what they mean when they say they sequence the DNA. Basically what they are getting is what you can see here in this section. Basically the reading of the whole sequence so that we can, by the time we get to the top of our gel, read the entire sequence from start to finish. We can also visualize DNA with dyes. This is ethidium bromide that is used on an agarose gel, and this is visualization by using radioactivity that's attached to the DNA before the gel has been run. Note that we can use basic gel electrophoresis if we've radioactively labeled the DNA or we've used a southern blot to detect it, to know how long our sections of DNA is. This is very clear here, and this slide illustrates 2 points. One is that the longer fragments are at the top of the gel and the shorter fragments are at the bottom of the gel. You can see that by this number of repeats on the side. So when we have 16 repeats, it runs much faster and goes through more of the gel than it does when we have 86 repeats. This is helping you to remember that the larger fragments are at the top of the gel, the smaller fragments are at the bottom of the gel. With regard to recombinant DNA, all you need to know are these 4 things in this slide. You need to know that there are selectable markers, reporter genes, a multi-cloning site, and an origin of replication. You need to know roughly what they do for you and why they are important in a cloning vector. Basically, whenever a question tells you that they are cloning the DNA, the purpose of that is to make more of the DNA and only have that specific gene in large quantities. We do that by putting the gene of interest inside a reporter gene. The new gene that's put in here disrupts this gene, so we can actually test for whether this gene is functional or not. If we got our piece of DNA we want in here, it's no longer functional. We can also tell if we have the plasmid in the bacteria by growing it on the selectable marker type media. So, for example, if our cells are normally sensitive to ampicillin, only those that are ampicillin resistant due to having the vector can grow. So the ampicillin resistance gene tells us that we have the plasmid in the bacteria. The origin of replication is used to actually replicate and maintain that DNA inside the bacteria. And the reporter gene provides us with the cloning site and gives us an indication as to whether we have actually cloned our gene properly or not. We can make probes in a variety of ways. Basically, we're going to go over all of these, so I'm not going to spend time on this here. One way we can make probes, or we can make large quantities of any DNA, is by the reaction called PCR. PCR, in short, is just a way to take a single copy of the gene and make many thousands to billions of copies of that gene in very defined lengths of DNA. The only purpose of polymerase chain is to make many, many copies of the DNA. The only other thing about polymerase chain reaction that you would need to know is that this reaction is carried out by a very specific DNA polymerase. That DNA polymerase is very insensitive to heat, so it is heat stable. DNA polymerase, the one that is used most commonly, is named TAQ, T-A-Q polymerase. We can also amplify what we have in nucleic acids, particularly from RNAs, and make them more stable. RNA is much more easily broken than DNA by using an enzyme called a reverse transcriptase and a sequence of DNA that is just deoxythymine, and make a copy of our RNA and turn it into a DNA double strand. Use that to clone it, or just use it as the probe direct. You need to know how to read and look at a dot blot. Remember, a dot blot is only telling you, yes, we have the gene, or no, we do not have the gene. For example, in this example that's here in this figure, they've probed the upper row with the gene for hemoglobin beta A gene, and they've probed the second row with the probe for hemoglobin beta S gene. This is the sickle cell allele. This is the normal allele. And so the first person here has a big red dot. That means they have hybridization. So they have the normal gene, but they're missing the red dot underneath, so they do not have a copy of the sickle cell gene. So they are homozygous normal. The second person is heterozygous because they have a normal hemoglobin A allele and a hemoglobin S allele. So this person is a carrier for sickle cell anemia. Likewise, in the third case here, we have none of the normal allele and we only have the sickle cell allele. So this person would be homozygous for sickle cell anemia. You need to be able to read and look at the color schemes for a comparative dot blot. These are often called microarrays. Microarrays use often two colors of fluorescent dyes. They use one cell that is normal and they put one color of fluorescent dye on it. They use a different cell that has a different fluorescent dye on it. Usually one cell is labeled with a green fluorescent dye, the other is with a red fluorescent dye. When you have equal amounts of both red and green fluorescence, you see yellow. If you have a bit more of the red in the comparison, then you will see it as orange. If you see it as a yellowish green, that means there's more green material there than there is red material. And of course, dark green means that you only have the green type RNA or whatever you have labeled, and red would mean you have only that you got from the red cell. This is just a reminder of in situ hybridization. This just reminds you that we can probe and we can actually paint the chromosomes, or we can actually look at specific regions of the gene to look at why those genes are there or not. Remember on restriction endonucleases that they always cut in a palindrome. This statement that they always cut in a palindrome is very important. If the palindrome is mutated as it says here, that restriction enzyme cannot cut. That will actually be the case when we look at some southern blots for sickle cell anemia. Restriction enzymes can have 3' overhangs or be sticky. They can also be blunt. These overhang ones are more conducive for cloning, but they don't have to have the overhangs, but they always have these palindromes so that if we read it, it reads TC and then the complement of TC, GA. Each of the restriction enzymes is named for the microorganism for which it was first isolated. We use restriction enzymes to look at variations in alleles because each person in a room like this has a slightly different allele and slightly different restriction enzyme cutting sites. This is a gel to show you that these bands are due to the same gene being cut at different locations. In lane 1, it's cut into 2 pieces. In lane 2, it's cut into a single piece. In lane 3, it's cut into 2 pieces. Lane 4 has 3 pieces, and there are these specific sizes that we can see here. How do you read? And then southern blotted. This is a southern blot of the gel. It could also have just been an ethidium bromide like we saw in the previous slide. We have a probe. That's why we know it's probably a southern blot. This is the MSTE2 site. The MSTE2 site is lost when the mutation to form sickle cell anemia occurs. The point mutation that creates sickle cell anemia just disrupts that palindrome. Now, when we look at this pedigree down here, we see people with the sickle cell have a 1.35 size fragment, but the people who have normal hemoglobin, hemoglobin A, have 1.15 kilobases fragment. So this person has both types of fragments, both sizes. So this person is a heterozygote. This person is a heterozygote. So both of these people are carriers for hemoglobin S, or sickle cell anemia. This person, however, only has the 1.15 fragment, and remember that comes from the normal allele, so this person is 100% normal. This person actually has sickle cell anemia. We can tell that by the fact that they are colored in the pedigree, but we can also tell because they hybridize to the 1.35 band, which is the sickle cell anemia band. They only have one band, so they must be homozygous. We can do a similar process by using the combination of restriction fragment length polymorphisms and the fact that we have repetitive DNA. These repetitive DNA sequences are called variable number tandem repeats. Variable number tandem repeats means that we have repetitive DNA off to the side of our gene, and we can use restriction fragments to cut around our gene of interest, use a probe to that variable region and tell how big that fragment is, and then we can tell what lineage the person came from. This is what I call the Who's Your Daddy type experiment, where we actually look at what is the number of alleles for each individual. Here the mother has an allele that has 6 repeats of that repetitive sequence, and in gene A she also has a second allele that has 8 repeats. She has 9 repeats in one of her gene B alleles or 10 repeats in the other gene B allele. So to prove that the baby is hers, she must have exactly the same number of repeats as the baby. So she probably gave her 6 allele to this baby, and she probably gave her 10 allele in gene B to the baby. If we wanted to figure out which of these fathers also were the father to this baby, what we would need to do is say, since the mother gave the 6, the father also has to give a 6. So he could be male 1, male 3, or male 4. For gene B, however, the baby got the 10 from the mother. That means the 5 has to come from the father, and the only father with a 5 repeat allele is male 3. This proves that male 3 is probably the father of the baby. This is more on rift-lip mapping and southern blotting. Remember, we went over this in class. The short form here is you need to look for changes in size of the DNA. If the DNA goes up in size, as it does in this diseased patient relative to the normal here for the lower band, that means there's an insertion. If it had come out much lower than the normal band, or as low as the normal band plus a little bit lower, that would have been indication of a deletion. If it is at the same size, then we assume it is the same band often. But you do not necessarily have the same band every time. In this southern blot, we might actually look at this and try to figure out which of these children have the disease and which are carriers. That would allow us to actually go through the genome, and we have to figure out who in the genome did each of the children get their bands from. Notice that in this case, the mother and the father both carry a 5.7 allele. But since the mother is considered healthy, if they get the 5.7 allele from the mother, that's probably a healthy allele. And if we get it from the father, it's probably the unhealthy allele. So this child got that 5.7 from both parents, so it must be homozygous for the 5.7 allele, but heterozygous with regard to the disease. Likewise, this child right here got a 5.7, but we need to figure out where it came from. It must have come from the mother, because the mother has a 5.7 and a 4.7, but she does not have the 3.2, 2.5 alleles. So that means that the 3.2, 2.5 allele, which is this top allele, must have come from the father. And he does have that allele as well. So the lower bands are from the father in this case, and the upper band from the mother. So this child has a good 5.7 allele. This child is also got a 5.7 allele and a 4.7, but since the father does not have a 4.7, we know that the mother must have given the 4.7 allele and the father given the 5.7 allele. On microarrays, what I want you to know is that microarrays are an up-and-coming technology. They have become more and more common in diagnostic testing for medicine, so you do need to know about them and how they are arranged. We can now use them to detect nucleic acids by hybridization, or we can use them to detect proteins and peptides, or even glycans or drugs. There's a wide variety of uses for microarrays. All of the microarrays have names that tell you what that microarray does. This microarray is called a SNP microarray. It detects single nucleotide polymorphisms. It's looking for point mutations. This kind, called a gene expression microarray, would be an array that is looking at the cDNA or the RNA of a cell and seeing which RNAs are expressed at a given time in the cell or in a given cell type. So it's looking for the amount or timing of a RNA or the spliced variants of RNA. You can read all of these and tell the various kinds of microarrays there are. Generally, on the test, all you have to do is read the name of the microarray and it tells you what it does. For example, comparative genome hybridization is comparing genomes and by hybridization. They come in various types. We can use a similar microarray to do multiple jobs. For example, this is the same microarray in both images. In this case, we're using genomic DNA bound to the microarray, and we're comparing how much DNA whatever we labeled in red has relative to whatever we labeled in green. And of course, where it's yellow, we have the same amount of DNA in both cells. This is a comparative genome hybridization allele. It tells us copy number of the genes because we're looking at the DNA from the whole genome. In this case, we're looking at an expression array. So we're looking at cDNAs made from the RNAs that were isolated from these two cells. And so we can see that in many of these RNAs, there's the same amount of RNAs in both cell types, but some RNAs, there's more of the red type, and others, there's more of the green type. A GWAS, or genome-wide association study, is just a technique we use to scan the entire genome looking for large insertions or deletions, as we did in this experiment. Basically, you take sibling pairs and you compare them to each other. The vast majority of the normal individuals fall along this static line. Anything that falls greatly out of that line for our patients that is different than the normals, we call that significant. This is very significantly lower than that, so that means there was less copy number of this set of genes. And so it tells us how we can determine how big the deletion is, and that most of the people who have this disease also have this deletion. We can do that when we're looking for multivariate diseases that have many genes that contribute to the onset of the disease. And again, you would use sibling pairs and then plot where the normals are and the average, even affected individuals are, but where the infected individuals pop way above the average, those are areas which we feel we might want to show more interest and figure out if they are truly involved in our disease. The last part of the lecture was on replacement therapies. We can do replacement therapies by giving our patient either replacement stem cells or replacement enzymes. The replacement enzymes would be the best in many cases, but the difficulties is getting the enzyme to the location within the cell that we need that enzyme to work. As of the present day, these are the only diseases that have stem cell and or enzyme replacement therapies that are approved for use in a clinical fashion in the United States. We have used many techniques for this. We have used viruses and a variety of nucleases to facilitate this. The problem with most of these techniques is they do not work well because they have the side effect of creating other diseases such as cancer. The only approved gene therapies at the present time are these four examples of gene therapy. We have tried liposomes, we have tried viruses, and we have tried nucleases to make gene editing work. You do need to know the difference between a knockout and a knock-in. The way we do that today is by a system called CRISPR-Cas9. We'll see CRISPR-Cas9 as a huge benefit over the old systems. These are the old systems. Both of these are rarely used today because they have such severe side effects. What is used today is the CRISPR-Cas9 system that utilizes an endonuclease called Cas9 that relies on a guide RNA that directs the cutting to a very long, highly specific region of our genome. The beauty of CRISPR-Cas9 is that in CRISPR-Cas9 you have this very long sequence. You can actually vary the sequence from about 20 nucleotides to about 35 nucleotides. At 35 nucleotides, you can actually be assured that you have exactly one region in the human genome that it will bind to. This guide RNA binds our target region of our DNA. It makes a double-strand break. At the same time, we either introduce another piece of DNA or we allow the replication machinery to try to repair this, which usually results in an interruption by an insertion or deletion of our gene. If we throw in donor DNA at the same time, we can actually cause it to do homologous recombination and fix our double-strand break using the gene that we give it. Thank you for your time. Good luck on the Mini. Hope these videos are helping you. I would love to hear from you if they are helping you so that I know that they are worthwhile to continue to produce and that people are actually using them and getting some benefit out of them. Thank you.